This video is over translation, which is the process of converting RNA into a protein. So, you begin with a strand of mRNA. Focus. So here's your strand of mRNA. And what happens is this mRNA has instructions for building a polypeptide through what are called codons. Codons are, like, for example, it's a three-letter base pair, basically. So AUG, that's the start codon for everything. So this little section of three bases is what's considered a codon. So what happens is your mRNA is inside a ribosome, which has its alpha and beta subunits. You have the E, P, and A sites, and so when the start codon, like AUG right here, reaches the A site, a tRNA molecule with the anticodon and an amino acid attached comes along and it binds to the AUG. So the anticodon is just the base pair that would bond together with it. So it would be UAC. So yeah, that comes over here, and now you've got UAC, tRNA, and it's got an amino acid attached. So next frame, same setup. Now you've shifted over one more section, so your start codon's now at the P site, and you have a new codon at the A site, which let's just say is UCG or something. And so you'll still have this tRNA here with its amino acid, and then the anticodon for this one right here is going to come over here. So it would be AGC. That tRNA also has its amino acid attached, and what happens at the P site here is that these amino acids form a peptide bond, and then this amino acid right here releases from its transfer RNA. So now you basically have two amino acids connected together. Now, whenever it shifts over one more, the E site just means exit, basically. So one of these tRNAs is going to exit, and you'll have another. This time, this one will be in the P site, and its amino acid will bind to the next codon's or anticodon or wobble, however you want to say it, amino acid. So you'll get, basically you're creating a long chain. You're creating a polypeptide, a ton of amino acids together, until it reaches what's known as a stop codon. There's a few stop codons. I didn't memorize them or anything, so I'm not going to say them, but it reaches the stop codon, and the stop codon is recognized by a protein called a release factor. So you'll have this here. Here's your tRNA. It's at the P site right now. And so a release factor protein comes in here and it fits perfectly at that P site. And basically what that means is that what it does is it kind of just dissociates the whole entire thing. So the release factor comes in, and basically everything just comes apart. So your mRNA goes off, your tRNAs are pretty much all gone, and then you have your polypeptide left, which is, you've made a protein now. That's what a polypeptide is. So that is translation.